Okay, good morning, everybody. This is Miss Torres once again. This is section 7 dash 3, the cell boundaries. In this section, we're going to cover how a cell transports material across the membrane so that homeostasis is maintained. And this is we're going to look not only at the structure of the cell membrane here, we're also going to look at the processes that the cell uses to get things in and out. So to get things out of the cell membrane and to get things into the cell membrane in order to maintain the homeostasis and the health of the cell. So, uh, what does it mean for a cell membrane that a cell membrane to have a lipid bilayer? So, lipid refers to fat. Bi the prefix bi means two, and layer is a layer. So, literally, it means that there are two layers of fat molecules that make up the cell membrane, and that's what you guys need to write down and what you need to know about the cell membrane, along with many more things that are part of the cell membrane. Okay, so this is a picture of the cell membrane, similar to the picture on the first section of this slide set. So here we've got a lipid bilayer that you can see right there, two layers of fat. They're the little balls with the two little legs. That's a phospholipid. And you see the green things. The green things are carbohydrates. They're carbohydrates chains. And you'll see the little green bubbles there. They say glycoprotein and glycolipid. Those glycoproteins and glycolipids is basically they start with glyco, which refers to the glucose or the fact that they're carbohydrate chains, and one of them is stuck to the lipid bilayer. That's why it's called a glycolipid. The other one is stuck to a protein, so that's why it's known as a glycoprotein. In the diagram, you'll also see the cholesterol labeled there. The cholesterol, basically, you can see how it's embedded between the membranes, and that's how it prevents the membrane from moving too fast when it gets hot and dissolving, and also when it gets too cold, it prevents the membrane from sticking together and freezing over. So it allows you to stay pretty much fluid and nice and the cell membrane to stay nice and bouncy even when it gets too hot when it gets too hot from dissolving and to when it gets too cold from freezing and making us like brittle and falling apart when it gets really cold. So you can go skiing and still not freeze over because of the cholesterol molecules right there. So that's actually what doctors call the good cholesterol. So you don't want to get rid of that. Then you've got the purple structures, which are protein. So those proteins are embedded in the membrane. Some of them are just stuck on the surface inside, holding part of what you see there, the cytoskeleton. And those proteins act as channels to get things out or bring things in or hold things down to the membrane, as you can see here. Okay, so here you're going to have to label all of this diagram. You have to label the carbohydrates there, the glycoproteins and glycolipids. You have to label the proteins, the fact that many of those proteins are actually opening. Some of them are anchors, as you can see there, they're holding down the carbohydrates. But some of them, as you can see there, the protein channels are little openings to get in and out of the cell. And it's noted, you should note that this layer is very flexible. So it can move around. It not only moves, some of them flip places. Some of them, it's just movable. It's constantly moving. It's basically the cell membrane. So, those phospholipids we talked about them in section 2-3. It's one of the types of lipids that we looked at under the macromolecule section that we covered. So, phospholipids are basically what the cell membrane primarily is made out of. So, it's that little round head with the two little tails. Well, this is what we know it's made out of. So, the heads are called polar. That's because they like water. They like hanging around with water. That's why they're on the outside. The two tails, are lipids. They're fats. They do not like hanging around with water, so they kind of just float towards the inside with other lipids where they feel comfortable. So they hate water. They all kind of hoard around the inside of the membrane. So it basically keeps this layer intact. So phospholipids, make sure you know their structure, make sure you know the polar head and the non-polar tails that they hate the tails are hydrophobic, which means they hate water, and like the word phobic is afraid of, they hate water, they're afraid of water. The head is polar, that means it likes water, or hydrophilic, so it likes water. Hydrophilic, philic means liking, and water, obviously water. Hydro is water, hydrophilic. So here we've got the cell membrane. Okay, so the cell membrane itself is called semi-permeable. Okay, so to permeate something, if you think of a sponge and if you put it in a big thing of water, the sponge will get all filled up with water. Well, that's what we're talking about. That's completely permeable. If you take the sponge out, it's going to be really, really heavy because all the water has just gone into it. It's completely permeable to water. Now, if you jump into a pool, you're not gonna like become really heavy and never be able to get out of the pool because you're so heavy with water. The water can't get in unless, of course, you decide to drink it all, in which case, yeah, you'll get heavy, but your body will start taking it out because you'll pee some of it out. So your body will try to get rid of whatever it can't handle. Otherwise, it just dies. And there has been a person that actually drank so much water that they killed themselves to win a contest a few years ago, a radio contest, and somebody actually drank so much water that they diluted their blood was basically so dilute that it killed them. So you don't want to introduce that much water. You have to give your kidneys the chance to get rid of some of the extra that your body doesn't need. So it is important to regulate what comes in. You don't want to jump into the pool and all the water just to come in you because it'll kill you. It'll dilute everything. Your blood won't be able to take care of business. Your cells won't be able to take care of business, and you'll die. So it is really important that your cells take up as much as they need and get rid of everything they don't need. So the cell membrane is what we call semi-permeable. So it must take in the proper chemicals that your body requires and get rid of those that it does not. That is really an important function of the cell membrane. And it needs to be semi-permeable because we don't want everything in and we don't want to let everything out. So it's kind of permeable. Some things in can get in easily. Some things have a little bit harder time. Some things can't get out at all. Some things can get in easily. Some things can get have trouble getting in but need a little help. And then some things cannot get into the cells. So it's called this is semi-permeability. It's kind of things can get in. Okay, so now based on your understanding of what semi-permeability is, think about that at a screen, window, or door, of a screen on a door or a window. How is that similar to how a cell membrane behaves? It's up to you to answer this. It's not going to pop up for you, so make sure that you answer this fully. It's going to be checked. The cell membrane is for protection and support. That is its function. So make sure you write that as the cell membrane function. And it's often referred to as a fluid mosaic because a mosaic, as you can see here in the picture, is basically made out of little pieces to make up a whole picture. And it's fluid because it moves. So what a mosaic is you see them at churches where you get the panels and it makes up an image. You see them in some forms of media where you get the little it's basically pixels. Pixels, you put them together, they make an image, and that's what it's basically that's a mosaic. That's what is referred to as a mosaic. So the cell membrane is really made up of little tiny pieces all stuck together. So the cell membrane is often referred to as a fluid mosaic. Okay, so this is a picture of a plant cell. I hope you can tell that because it has a cell wall and it has a tons of organelles and a giant vacuole. So here we've got a plant cell, and once again, they are different because they have not only the membrane, they also have a wall on top of that. So I want to talk a little bit about the cell wall. So, cell walls are present in plants and most prokaryotes. Most bacteria have it, but some of them don't. There's a few that do not have walls, but most do. The cell wall is right outside of the cell membrane, and it does have pores and openings that allow things to pass through them. So they can get through. First has to pass through the wall in order to get to the membrane. So items need to get through the wall to get to the membrane. Now, if you've ever watered plants, you know the fact that if you try watering just the leaves, but it doesn't get the roots, you're not watering the plant at all. Because plants have the top parts generally are for different purposes. They don't have a lot of openings that allow water in. So they're pretty strong, that cellulose, and the water can't get through them. And some of them have really waxy layers that basically prevent water from sticking on them. So some cell walls are not don't have pores that allow water in. So those pores are found somewhere in other parts of the cells, like the roots. So those pores are through cell walls are generally only located in certain areas, and for good reason. I mean, if a plant had pores for water, the water can come in and out equally. It'd have a big problem, especially on hot days. So make sure that you understand that plants and most prokaryotes have cell walls and they are outside of the cell membrane. So they still have a membrane, they just have a wall on top of that, an extra layer of protection that does at times let things in and out because they're spores. So once again, the cell wall is for protection and support of the cell. Uh, Animals do not have cell walls, they just have cell membranes. Plants have both cell walls and cell membranes, and the cell wall is made up of cellulose. So please remember that cell walls have that plant cells have both plant cell walls and cell membranes. They just have an extra part. So this one here shows you the movement through the membrane. Okay, so what we know is that water especially moves from where it's more concentrated to an area of less concentration. You could see that with the arrows. And water generally is small enough to move through the phospholipid layers and get itself in. You could see that right there. So water can easily make it in. Larger molecules, you could see like these sugar molecules you're labeling here, cannot get through the membrane. They are prevented from moving through the membrane because they are too big. So we're going to talk about how water moves first of all, and then we're also later on going to talk about how we get those large things that our body needs into the cell. So we're going to talk about how those things that can't pass through the membrane get through the cell membrane. So there's basically A and B here. There are the cell things get into the cell through two processes: active transport and passive transport. Active transport requires lots of energy. Passive transport requires no energy at all. So no energy is required for passive transport. And there's three ways of getting through the membrane without using any energy. They are simple diffusion, facilitated diffusion, and osmosis. Active transport is pretty much an energy requiring pump. So you're using a pump, and when you use a pump, any pump, even the ones that people use, like a vacuum to get the water out, those pumps require energy. But we're going to talk about those after. We're first going to address the simple diffusion, the facilitated diffusion, and osmosis. So, passive transport, once again, is simple diffusion, facilitated diffusion, and osmosis. We're going to talk about the difference between these three methods. Okay, so simple diffusion is simple. Basically, particles move from where they're more concentrated to a less concentrated area. If you know someone that wears like a ton of really annoying perfume, you'll see that the smell kind of that where they spray it, and let's say they bathe, it seems like they bathe in it because they put so much on them that it's like they walk into a room and it just kind of starts moving, and pretty soon everyone in the room can smell it. Well, it's the same thing. This is just simple diffusion. Particles moving from where they're more concentrated to where they're less concentrated. We see this in the human level. We see when you're at in a crowded area, you tend to try to move to areas where that are less crowded. During lunchtime, I see people doing that all the time. The quad area gets really crowded, so people try to move over here towards like these areas over here towards this building because there's so many people over there. People like moving from more crowded to less crowded too. So we not only see particles moving from areas that are more crowded to less crowded, we see that happening in humans. I see that a lot. So things moving from a more concentrated area to a less concentrated area is a very natural thing to happen. Okay, as you can see here, there's an impermeable barrier in one of these. Impermeable means that nothing can get through. And as you can see there, none of those molecules are getting through because it's impermeable and they cannot move through. Then on the other one, we've got a semi-permeable barrier. It's got little openings that let some things through, but not all things. What's moving through? you can see the green molecules moving through. Not the red ones can't fit, so they don't go from high concentration to low concentration. So on the first one, the impermeable, there's high concentration and low no concentration on the other side. And in the semi-permeable barrier, you see this high concentration to low concentration of only the green ones. The red ones are not moving at all. They're just staying at a high concentration on one side. So generally, we get a movement until the state of equilibrium is reached. Equilibrium is when the concentration of the solute in the same is the same throughout the system. So the concentration of the solute is um, is the same throughout the system. That's what we're looking at when we're talking about equilibrium being a balanced state. And you could see the picture on the right. The green ones, there's kind of equal amount of green and green on one side than the other. They're kind of even. The green ones are at a state of equilibrium. The red ones are not, but the green ones are. So they went to equilibrium. And generally, that's when simple diffusion stops, when, or kind of gets to that even level. We call it equilibrium, not the same, because equilibrium means that it's moving. There's still some green ones moving back to the one side, but the other maybe another green one's going to the other side. So it's kind of moving constantly. We're talking about a liquid, so things are moving around a lot. Okay, so simple diffusion. Is it specific or precise or particular or general, and why? Simple diffusion is general because as long as something is small enough, it can spread wherever it wants. It just kind of goes from high concentration to low concentration. If something's small enough, it can just move from high concentration to low concentration because it's small enough. Sometimes it's a good thing, like water moving from high to low. That's a good thing. But then we get things like there's lots of molecules that do this from high concentration to low concentration constantly that we do not want in our system, and that would be really bad. So make sure that when you go from high to low when things move from high to low concentration, if they're small enough, they can do that just randomly whenever they want. So they're able to go from high to low concentration. We have a couple of things in nature that would be really poisonous. So things that if you can get like for example, nicotine. Nicotine is a really small part made out of really small particles. So if you put it to your skin and touch yourself, you can die from nicotine poisoning. That's why people smoking, getting nicotine into their system through smoking is really super dangerous because nicotine is highly poisonous. If you get the liquid nicotine and put a drop on your hand, you will die. It's very toxic. So it's really strange that people actually want to smoke it. But the fact that you can put nicotine, just a drop of nicotine on your hand and it'll kill you is really bad because it just goes into your system and travels around and basically messes up your entire system because it's very permeable. It can go from high concentration to low concentration very easily, and we try to avoid those things. Facilitated diffusion, the second type of passive transport, requires protein channels to allow the molecules to move across the membrane. So these are molecules that are bigger, so they can't get through the cell membrane easily. So they need these little doors to allow them to go through. So you see here in the pictures, like this little Pac-Man shape, that's a protein on the membrane, and it basically just grabs a molecule. It doesn't suck it in, it doesn't use any power, but basically once a protein gets in there, it triggers it to close it, and then it opens on the other side, allowing the molecule to go in. Okay, so that is essentially what happens. So why do we need facilitated diffusion at all? Why can't we just use simple diffusion all the time, allowing molecules to just get in as from high concentration to low concentration until they reach equilibrium? Well, the reason is because some molecules, like glucose, are too big. They can't get through, yet glucose is our main source of energy to run our cells, so we need those things to get through. So they need protein channels instead. Some molecules just don't get along with the membrane, so you have the little, they're actually they're made out of fat, and like the outside of the membrane only likes water things, so it needs a different way to get in. So those things need to go through special channels. So things that require that aren't able to get to simply travel through the membrane because of their size or if they're the wrong chemicals, they and our body needs them, they move in through special channels. Now, as long as something's moving from high concentration to low concentration, it does not require energy. So just to remind you, facilitated diffusion does not require energy, just like simple diffusion. No energy required. So facilitated diffusion, here you have a diagram that you have to label, and you see the high concentration on the outside of the cell of these square molecules, and you have the phospholipid layer and a phospholipid that you have to label. You also have a protein channel, and you see how it's moving from high concentration on the outside of the cell to inside the cell where there's less of the concentration. So you see this is kind of a progression. You have the first one where it's taking in the molecule, and that's triggering the molecule to basically swallow it and then deposit it on the inside. So as long as it's moving from high concentration to low concentration, none of this requires any energy. So, is this very specific or precise or particular or not, and why? Well, the reason this is actually very specific because only certain substances actually are able to do this. As you saw on the previous slide, it has to have the right shape to trigger it to go inside. If something triangle tried to go into that same protein, it would not swallow it. It wouldn't trigger the protein to swallow it. It just kind of would bounce right off. So once again, this requires what we called active site. The only way you trigger that protein to take that molecule in is if it has the right shape or the right active site to trigger the swallowing of the protein of the protein swallowing the whatever chemical it is. So that protein will only allow those things that match its active site to be taken in. So it is actually very specific. So facilitated diffusion only allows things in that are big that match the right shape that that the cell needs. So, passive transport Venn diagram. So let's compare simple diffusion to facilitated diffusion. So, which one's general and which one's specific? So simple diffusion is general, facilitated diffusion is specific. Uh, in simple diffusion, the molecules move directly through the phospholipid bilayer. They're small and they move right through the bilayer. In facilitated diffusion, they have to go through protein channels. So those are needed in order for facilitated diffusion to work because those molecules are really big and they need something to swallow them into the cell. But both of these have things in common. Everything that moves through both of these processes is moving from high concentration to low concentration. And as long as something is moving from high concentration to low concentration, no energy is needed. No energy is required. So make sure you understand that both of these processes require absolutely no energy because of the fact that molecules are moving from a more crowded area to a less crowded area, and things just do that naturally. So transport, simple diffusion, and facilitated diffusion. This is just a different way to look at the same information from the Venn diagram. Simple diffusion, small substances that can diffuse across the membrane. It's very general, no energy is required, and that's because everything moves from high concentration to low concentration. Facilitated diffusion, that's for molecules that are too big to cross the membrane, so they need proteins to swallow them and take them through. But this process is very specific because we don't want other big things that may harm your cell to get through. No energy is required for this either, so movement through this has to be from high concentration to low concentration. Okay, so this is a little trickier because during this process, you see the first one you see there, you could see that there's a higher concentration of water on one side, and on the other side, there's less water. So you start off with one side that has a lot of more water than the other side. But if you look at those molecules there, let's say those are sugar molecules, the ones in kind of purplish there. So the purple molecules there's less purple molecules, so that's called the solute. There's less solute on one side, less purple molecules on one side, and more solute or more purple sugar on the other side. So, and you have there a selectively permeable membrane. If you look at the little openings on the membrane, the big molecules, can the solute move from one side to the other to where it reaches equilibrium? And if you look at it, if you look at the semi-permeable membrane, you know that the large molecules cannot move one side to the other. So we snap over to the other side. So over time, what happens? Well, the things that can move are water molecules. Water molecules can move through that. So because there's less water on one side and more water on the other side, so there's more water on one side and less water on the other side because of the amount of sugar that's in on one side. The water moves to where there's less water. So water moves from the higher water to the less water. So it moves from the left to the right side, making one side of the U-shaped tube higher, to look higher. But if you look, now the solute is even on both sides. So the water moves to where the water becomes equal. And by default, that makes the even concentration of the solute. So one side now has about the same amount of solute per compared to water than the other side. So if you would think this in practical terms, you started off with one side. Let's make those molecules Kool-Aid. Okay, so one side has a little bit of Kool-Aid molecules and a lot of water. So really, it didn't taste like Kool-Aid. It tasted more like water. And the other side was really super, super sweet and it had tons of Kool-Aid. Okay, so one side is total Kool-Aid, the other side it doesn't even look like Kool-Aid because there's so little Kool-Aid on it. So then the water then moves to the other side. Now both sides taste this. One side kind of tastes less like really strong, and the other side tastes more like Kool-Aid. So now like the Kool-Aid can't pass through, the Kool-Aid molecules can't move through, but the water did. So now you've got equally tasting Kool-Aid. Now on both sides it tastes like the same. So the water moved to make the concentration the same on both sides. So now one side isn't totally like super, super thick because it has so much Kool-Aid, and the other side doesn't taste like water. Now both sides taste just about right. So that's what we call osmosis, water moving from higher concentration to lower concentration. So the water moved from where there was more water to where there was less water. Please take a look at this because this is actually a pretty difficult concept. It looks relatively easy, but it's a really difficult concept to understand. And in class, make sure you ask plenty of questions about this. Okay, so active transport. Active transport is like implied in the active, it requires energy. So substances are moving from where they're less crowded to where they're more crowded. In order to move something and kind of pack it in, you think about how much energy you need to put something into an already packed box, or add more things in when it's already full. You need a lot of energy to push it in. It's kind of like I can think of like when you've already got like tons of stuff in the refrigerator and you're trying to put something else in to the refrigerator. It's harder to find a place to put everything, and you may need to rearrange some things to put fit in the thing you need to put into the refrigerator, as opposed to when there's nothing in the refrigerator, you just kind of put things into the refrigerator. There's lots of space. So we're talking about moving from where there's less concentration to where there's more concentration. So from less crowded to more crowded. Diffusion against the concentration gradient is officially what we call this. So moving from low concentration to high concentration. So, active transport requires this use of this ATP molecule. This ATP molecule in the red circle there basically captures things and takes them in, sucks them in. So it's not like it's kind of like passive transport, passive facilitated transport where you have a protein. But this protein doesn't just swallow the thing. It does swallow the thing, but it requires energy to suck them in. So these proteins are not only swallowing the thing, they actually suck the things in whenever they get caught. So they're like pumps. They pump the thing in. They first have to like suck it in. That's what they're doing. And in order to do this, you need energy. So that's what that ATP is. We're going to talk about that next chapter, adenosine triphosphate. So active transport uses carrier proteins. These are protein pumps and they require lots of energy. So they suck things in. You can see this protein right here. The things the actual molecules are being sucked in, and every time they need energy just to bring things in because they're going from less concentration to higher concentration. So This picture here, you have a worksheet to fill this out. You've got the two at the top, the little circles are able to fit through the membrane. So they go from where they're more crowded to less crowded. This is called simple diffusion, and that's passive transport. You've got these triangles here you have to draw. There's a whole lot of them out here, and there's a little bit inside, but they don't fit through the membrane. But they do have a protein here that they can go in, and as long as they're moving from high to low concentration, that's called facilitated diffusion because it does need a protein, and that's called passive transport. You have to draw in your protein there to and the triangles and the arrows and label it facilitated diffusion. And still high to low concentration. So as long as you've got like the two at the top that are moving from high to low concentration, that those are called passive transport. And then we've got here um, these little green squares are being sucked in from low. There's only two of them out here. You see them? And they're being sucked in by this round purple protein pump. But to run that pump, you need energy here. You've got ATP looking like a little lightning bolt, and that little energy burst allows that protein pump, to pump them from low concentration to high concentration. So these things are being forced in by that protein pump, and that protein pump requires energy or ATP. So that protein pump is powered by adenosine triphosphate that allows it to pump things from low concentration on the outside to high concentration on the inside, and that's called active transport. Okay, make sure you have all of these clear and know how much they use.